So I guess good morning, everyone. So thanks for coming so early. I know it's 8:30 is the time for a primer. So my name is Shamil Sunyaev, and I will be talking about primarily annotating genetic variants and what is functional, non-functional in the genome. Given this is MPG, I will try to start with the genetics perspective and when does the issue of annotating gene variants arise in genetics. And please interrupt and irregular MPG style, so stop me at any point if something is too simple and boring or something is unclear and requires a little bit more detailed explanation. So again, from genetics perspective, you may remember from your genetics class that about 100 years ago there was this debate between biometricians and Mendelists. That Mendelists suggested that this is what genetics is, so there are traits that nicely segregate in pedigrees according to Mendel law. And biometricians said, no, this is all an auditing, and what's really interesting is that if I look at any population, for example, fish in the pond or corn in the field, what I observe is variation in certain traits. Many of them are quantitative, and there are certain statistical laws that govern inheritance of these traits. So what's happening today is we don't really know, but we treat our genetic annotation of individual alleles differently in Mendelian world and in complex trait world. And there is a little bit of dichotomy, and many people think that this is not a real dichotomy, but I'll start with discussing annotation from this perspective, and then we'll move on and maybe at the end discuss whether this makes sense or not. So if we're dealing now with a Mendelian trait, so we have a Mendelian trait, we have something which segregates in the pedigree, would like to analyze it. Of course, long time ago, people did linkage studies and found loci segregating in the population and doing sequencing in this locus and trying to find a hit. Now you don't really need a large pedigree anymore, and frequently it is sufficient to work with a trio. And in many cases, you have unaffected parents and an affected child. And if somebody tells you in this case that the phenotype is monogenic Mendelian phenotype, which is not always given because you don't have any segregation information in this family, you can think, oh, if this is a fully penetrant variant, it can be either dominant or recessive. If it's dominant, it means that this mutation occurs de novo because none of the parents is affected. And what we know from population genetics, number of coding de novo mutations per generation in humans is limited. It's on average about 1. And it's a textbook Poisson distribution, so you can get 0, you can get 1, maybe 2, maybe 3, very rarely more than that. So you end up with a small number of variants, and you believe that these variants are a very large effect, and they usually, we don't really know, but the community makes a bet that those are in the protein coding region of the genome. So the focus is on protein coding region of the genome. So what you'd like to do, you'd like to select one truly functional causal variant out of very few. Another possibility, this can be recessive inheritance. So it can be either homozygous or more frequently compound heterozygote variant. Again, you think that this is a highly penetrant couple of variants of very large effect. And if you remove everything you observe in public databases with substantial frequency, you end up with very few, 3 to 5, maybe compound heads, sometimes 1 or 2 homozygote variants, dependent on the pedigree. So again, what you have to do, you have to make a choice between very few variants, you bet that they are coding, and you assume that they are rare. Now, in complex disease genetics, and I understand that there are probably many lectures of this primer series about complex trait genetics, the whole situation is very different, right? So we can measure some phenotype, and there are certain statistical rules that govern its behavior. For example, many of the quantitative traits have distributions close to normal. And the phenotype is heritable, but heritable in a very different sense from Mendelian inheritance. Probably most of you heard about Galton plot. So what's happening here is that we can select mid-parent height and plot height of a child and observe that the distribution is approximately bivariate normal, that generally children of taller parents are on average tall, children of shorter parents are on average short, so there is a dependency. And this is why we think this trait is genetic. And then there are different statistical laws. For example, what they call regression to mediocrity, when you have children of tall parents are not as tall as their parents, and children of short parents are not as short as their parents, and so forth. So there are all these statistical rules that govern that. And what is biological function underlying it? How does this work? Again, this was a long debate between Mendelists and biometricians, and this was resolved by this paper by Fischer in 1918. And at this point, what I tell students in my class, there is a difference between science and humanities, right? So if you are a student of humanities, you're expected to read Aristotle and all the classic texts and so forth. If you're a student of scientists, you usually read the latest textbook and a bunch of papers published a couple years ago, where all of this historic knowledge is being packaged and prepared for you. So I think this paper is worth reading time as a classic text. It's a beautiful paper with, and the main issue why this paper is important, theoretical model of quantitative genetics is still what was developed by Fischer in 1918. Very little breakthrough since that paper. So what Fischer showed is that if we observe this variation in a trait, quantitative trait, we assume that there are many different loci, and by the way, they didn't know that DNA existed, none of that, right? So all of this was the model, which happened to be correct. There are loci, which he called quantitative trait loci, that influence this distribution. There are many of them, some of the alleles increase value of the trait, some decrease value of the trait, and each locus segregates in exactly Mendelian fashion, right? So now it's very obvious for us that a nucleotide variant would segregate in the Mendelian fashion because it's a discrete object. It was not obvious for Fischer and for geneticists 100 years ago because they didn't know what is the physical basis of inheritance. And the phenotype is additive, or you can put some interaction term over many, many locus effects. So you predict normal distribution, and then you predict regression to mediocrity, Breeder's equation, all of the biometrician statistical observations, right? Okay, now the problem we're facing with this type of inheritance in terms of functional annotation. So because there are many variants, and the effects are supposedly relatively small because each variant contributes to the distribution, we're not doing what we're doing in case of Mendelian genetics. We're not going after large effect, coding variants, and we have to select one among very few because we believe there is one causal variant, right? Here, the whole problem is statistically completely different. There are many papers trying to estimate how many variants and what are the effect sizes. A lot of this literature came from evolutionary biology and agriculture. And early papers were focusing on artificial selection experiments. So this is one I like because I cannot believe in this experiment, and I tell everybody about it because I read it in review, I checked the original paper, numbers in the text correspond to numbers in the figure, so it seems correct, right? So of course the easiest model to do artificial selection in animals is Drosophila. You can select for any trait, so this is selection for speed of flight. So what people did, they took flies and they took banana and they released flies and they fly towards banana, right? And average fly, they measured flies about 2 centimeters per second. Then they select flies which reach banana first, breed them, and introduce a fan, blow wind in the opposite direction. So the flies now have to battle the wind. Some of them can reach the banana. You take these flies, breed them, take next generation, ratchet up the speed of the fan, and flies have to battle stronger and stronger wind, so you're selecting for speed of flight, right? Now, what happens if there is a 1, 2, 3 loci underlying speed of light? You would see fixation of one allele, second allele, third allele, and there would be saturation. There would be no response to artificial selection. Right? So what's happening here is that you start with flies flying 2 centimeters per second, and you eventually, there is continuous response. There are 2 selection relaxation steps until you reach flies which fly almost 2 meters per second. I cannot imagine these flies. It's very hard to believe. I have no idea why Department of Defense doesn't fund research in Drosophila genetics, but this is the observation, right? How can we reconcile this with functional effect of allelic variance? What is happening? It seems that there are 2 possibilities. One possibility is that additive variance in the trait, so variation in the trait, is explained by multitude, large multitude of variants of very weak effects. So potentially the effect is statistical. So those are not this protein truncating rare variants we discussed in the Mendelian case, right? Another possibility is that you pick mutations on the go during the experiment, which also would point to very large mutational target. So the availability of a very large number of variants of potentially intermediate or weak effects, right? So this is the model, and this model is very different from the Mendelian genetics model. Now, a few years back, and now I think this is a hobby horse in human genetics, in animal genetics, hobby horse of many colleagues in our community here. There was this idea to try to fit models, in this case random effect models, so models which incorporate very small effect of very large number of variants. So this paper, for example, this is the pioneering paper in this, introduced infinitesimal model, meaning that you have distribution, every single SNP is important in case of height, and distribution of the effect sizes is normal. Right? So then I found myself in a difficult situation because you're thinking about function of variation, function of genetic variance underlying phenotypic variation. You go to one meeting and people say, oh, we have this family and this variant breaks this hydrogen bond in this protein and protein doesn't function and there is experimental set. And you go to a different conference and people say, oh, okay, so we start with a model which is infinitesimal. Every single SNP in the genome has some effect. The effects are very, very small. Right, so there are 2 completely different worlds. Now the good news is that the model happens to be stable, so you don't have to assume that every single variant is functional. It has to be certain fraction of variants, but still a pretty large number of variants with large effect. So what do we think is happening? So why would we have multitude of variants of a statistical relatively small effects? And here the focus of the community shifted in the analysis of variant function from this coding variance, what is happening biochemically with proteins, to this idea of regulatory variance. So there are, of course, many functional sites in the genome which don't code for any proteins, but collectively govern gene expression and potentially maybe some other functions we have no idea about, because again there are these conserved regions of the genome which apparently don't govern anything, we know. But because we at least know about gene expression, so this the focus has been on this variance involved in regulation. So before we move to actual annotation of function, a couple more comments. So we have this weak variance of many variants of weak effect model. We have this model of large effect protein coding rare in the population variants, and they are rare because selection kicks them out of the population and so forth. Do they point to different types of biology? And at least in some phenotypes where there was a lot of research in both arenas, this comes from a review in Cell by Sekathiresan, there is a very good overlap. So if you would look at genetics of lipids, you take loci which cause Mendelian phenotypes involving lipids, hypercholesterolemia, sorry, and Tanger disease and so forth. So you would see that this Mendelian loci and loci recovered by genome-wide association studies using common variants of weak effects, many of these loci are exactly the same loci. Then Jonathan Cohen and Helen Hobbs sequenced a whole bunch of protein coding genes in Dallas Heart study, looking for rare coding variants involved in variation in lipids, and many of these genes have effects of rare variants in their studies. So it means that even though we use variants of different allelic effects, regulatory encoding, genetic studies end up finding the same biology. Now, we don't know yet whether this is true throughout phenotypic space, but at least in case of lipus, this is the case, and many of those genes, this is reverse cholesterol pathway in case of HDL, same exact genes are being found, and they have clear biochemical functions, right? So both types of allelic variants point to the same pathways and networks and clear biochemical modules in cells. Well, in this case, not in cells because this is a transport. Okay, so this was genetic motivation, an introduction of the idea that we have to annotate alleles, and some of them would be very large effect, important rare variants, but others would be potentially common variants of very weak effects. Now we're facing with the annotation problem. So we have variants, and imagine even very simple Mendelian situation where we have a mutation in the family, patients. So how do we prove it's functional? How do we prove it's causal? So sometimes we have segregation data. That's a powerful mechanism, but it can work only if you have very large pedigrees, if penetrance is very high, if nothing sits on the same haplotype, so you assume that nothing can co-segregate with your variant. So in practice, this is being used and probably is the most informative type of analysis, but frequently unavailable. You have direct functional in vitro experiments, and this is frequently problematic because you may lack necessary assay. So for many variants, you don't have in vitro assay. If you do, you don't know whether you're measuring phenotype. Your in vitro assay completely recapitulates what's important for human phenotype, right? Because many functions of many proteins are pleiotropic and so forth. Animal model is great, but again, it has to be available, and there should be some assurance that animal model recapitulates human phenotype. Of course, frequency in the population controls is an informative, very highly informative feature. But again, if you have a variant which is very rare, even de novo mutation, there's no guarantee that it has certain function, right? So of course, if prevalence of your phenotype is 1% in the population and allele frequency is 50%, and you assume complete penetrance, this cannot happen. But if the penetrance is partial, or it's a small effect variant, or in case you'd like to have the opposite inference, that the variant is functional, this may not be very important. There are some bioinformatics approaches, and partly this is where part of my lab is in this space, and I think we're in business not because our methods are great, but because the need is so severe, right? And I'll talk about that in a bit. Okay, so you're facing with, yes, sure.
Can you just back up, because this is a very important point where I caught it here.
Yeah.
We have a number of genes, 20 or 30, that we've identified that cause absent puberty, isolated gene or deficiency. We go to the control population and we find many of these variants, and we have incomplete penetrance within the families, and we've documented oligogenicity. So we have multiple rare variants that synergize in 15% of them. So this is a really problematic thing when we go to Daniel's 16,000 or 90,000. We find these. So we have a burden test, which is kind of an artificial way of dealing with that. But it depends a lot on the bias of which controls you're using and how you accumulated your severe cases in the first time. So it's not a perfect analysis of these things. But everything on the right is tough sledding for the reasons you just said. And so bioinformatics would be a lot better if it could help us with this a little bit more. And I know you're about to embark on how good that is and isn't, I hope. But this is the thing, the incomplete penetrance is the bear here, because that's the rule. It's not the exception. I mean, the highly penetrant, rare things are extremely rare. Everybody has incomplete penetrance. And that gives you the problem with the controls. It gives you the problem at each one of these levels. Now, is there a way around this that you can start to deal with? This may foreshadow the rest of your lecture, but this is exactly where we're stuck right now. Even with 93,000 controls, we're turning up more of these rare variants in the population. And even more when we go to population studies where we have SNPs. Now we also turn these up and we see pleiotropy of these genes like cricket and other people. But it's a mess right now, and these two right upper quadrants are not there, and they're expensive, they're not scalable, they're not robust, they're not reproducible, they're not fast. So bioinformatics, it turns out to be the critical thing we're all relying on at this particular point.
Right, so no, I completely agree with you. So the situation where we are, and this is back about dichotomy, right? So you go to one meeting of Mendelian geneticists and everybody assumes complete penetrance, and everybody says, oh, this is not real mutation because I've seen it in a set of controls. So there are a bunch of papers now, some of them from our community here, where control population was sequenced. Apparently, clinical grade pathogenic variants were discovered. These people were brought to the clinic, rephenotyped, they are fine. So Cricket had a paper like that, Cricket Seidman, David Altshuler had a paper on Modi with Jason Flanack. So what is happening? So there are several...
It depends on how you phenotype.
Yes, that's exactly, but that's... Well, in Modi paper...
Lots of deep resequencing, we don't do much deep rephenotyping to parallel that. And by deep rephenotyping, I mean don't just look at how they lie, but give them a glucose tolerance test, stress them, look at insulin levels, go to the next level of pathogenesis. It's number one, expensive, and the issue of recall is a problem. But I wonder how many of those things, if you do those, are actually going to show.
Right, so what you're saying is, okay, so there are three possible answers, right? So one is that the statistical inference, the original inference about causality is incorrect. Correct. Those mutations are neutral. Another possibility is what you just mentioned, expressivity. So the alleles may be fully penetrant, but the effect, the phenotypic effect can be very variable. And the third possibility, of course, is that there is this huge dichotomy between fully penetrant variants and variants, many variants of weak effects included in complex traits, but there is a full scale of penetrance. So there are many variants of relatively high but partial penetrance. And this penetrance can be due to oligogenic inheritance. This penetrance can be due to epistatic interactions. There are many possibilities. It can be due to gene-environment interactions and so forth. So we don't know what penetrance is. We have no idea how to prove causality of these alleles, even if they have high penetrance but incomplete. We don't know what does this mean biologically. I'm in complete agreement with you. It's very difficult to get out of this. So I'll try to...
how do you write a grant about it to get a little more practical? Because no matter which reviewer you have, somebody doesn't like it. I mean, the Mendelian guys,
that's correct.
Guys.
Yes, that's correct. So there are opinions and there are strong feelings about the question. What is causality? What is functional effect? What is penetrance? I heard some people saying that if genetic variant is not fully penetrant, you cannot use the word causal. And other people are saying that, oh, we have this infinitesimal model, 10,000 variants are causal for this trait and so forth. Well, you just write that. It's a very tough problem and you're trying your best. I guess that's, that's what we do. Okay, so you're faced with this alleles, and what you do, at least in bioinformatics space with the annotation, I would quickly go through a variety of functions and where we stand. And the easiest and cleanest situation is where you have variants, for example, which are truncation variants or protein altering variants. But the important thing here is that every time you run the analysis, there are multiple transcripts and conflicting annotations, and this is very important. And when you look at any annotation of real data, you should follow conventions used by different labs. Because if there are always alternative, not always, but frequently alternative transcripts for a gene, and some labs follow the convention of the longest transcript. Others would follow the convention of most abundant transcript. Another possibility is use the worst case annotation if there is at least one transcript which may not be real, but the variant is predicted to be truncating, then this is the annotation you should follow. And a lot of discrepancies in real world are caused by simple conventions which are very difficult to find, sometimes in supplemental papers, and where labs are different. So you take nonsense variance as the simple, so stop gain variance, for example. Also, you can think about stop loss, frame shift, and so forth. And usually people take them granted as a clear loss of function variance, and functional interpretation is very simple. Now the problem is first that some of them may not cause nonsense-mediated decay. Some of them may fall in exons which are spliced out in common isoforms, so have no effects. And again, what's important is that nonsense variants are enriched in sequencing artifacts. So in whatever project you see, you measure fault discovery rate is greater for nonsense or loss of function variants. Why? Well, because there are very few, and the sequencing error rate is the same. So the fraction of errors among your finding is just greater. That's purely statistical issue. The next type of variants are variants involved in splicing, and variants involved in splicing, there are variety. There is one variety which I've seen a few examples is impossible to find: creation of a new splice site. So near perfect splice site somewhere in the intron, mutation causes makes it a perfect site. I have no idea how to develop method to find those routinely, just because they can be pretty much anywhere. Then we have conserved donor and acceptor sites, and that's relatively easy. Again, what's important to note is whether your exon is constitutively spliced or not. And then there are many splice enhancers and many variants influencing splicing which do not fall in the in two nucleotides conserved in donor or acceptor site. We try to rely on collaboration with a welfare brother, and I know there are other tools. We use a method called Spliceman, which is entropy-based method. So looks at sequence context and also uses a lot of heuristic tools, checking whether, for example, splicing out this exon would change frame and so forth. So there are certain bioinformatic tools helping with the annotation of splice variants. Important part here is that this is one type of variant. If you're willing to do some experimental work, mini-gene assays allow you to do the work without access to patient material, right? So you know the variant, you introduce the construct, and you experimentally measure whether it affects splicing or not. Missense variant, and I'd like to spend a little bit of time here, first because this is a big interest of us in the lab, and I'd like to spice it a little bit with the original research, but also because this is the most common type of functional variant in protein coding genes. And unlike splice variants and nonsense type of variants, where most of them are functionally significant, missense variants can be completely benign. Many of them, most of them are probably completely benign. So just changing amino acids anywhere in the protein. So there are tons of functional assays, bioinformatic methods. We usually look at evolutionary information, structural information, annotation. So I'll mention this in a second. Trying to predict functionality of the variants. So one tool a lot of people use, and I'll talk about how good this is, but mostly how bad this is comparative genomics or comparative sequence analysis. So you look at evolutionary information for the annotation. So this is the story, right? So you have your protein and you have certain site, and you can align other sequences. You can use paralogous sequences, but now with a lot of comparative genomic information, most people would use orthologs. So you have what we call the same protein from other species, right, which is orthologous protein. And you can look at the site, and of course, These are not independent data points, they are related by phylogeny, which has to be in the core of any statistical method, some molecular evolution taking phylogeny into account. The other problem is that there are 20 possible amino acids at the position, and you usually have too few sequences. You don't have enough sequences to go through all possible variation, right? So you have to be, statistically the methods have to be Bayesian. There's no way out of that. So what kind of assumptions these methods are making? And the main assumption, and I'll try to spend a little bit time of that discussing it, is the assumption of constant fitness landscape, right? So what we call in evolutionary terms constant fitness landscape. So what it means that if I see a variant and I assume it is good for fish, it's good for me. If it's bad for fish, it's bad for me, right? This is not necessarily granted. First, because I don't live in the water and I can give MPG talks and fish cannot. So we have very different environments. Also, fish has a very different genetic background. So if there are any interactions with genetic background, this assumption is not granted. However, if we make this assumption, then we can estimate two things. First, we can estimate whether the mutation, our human variant, fits the pattern observed throughout the evolution. And we can estimate the rate, so what a lot of people call conservation, at the site. This is a very difficult statistical problem. The current models used by the field are a continuous time Markov model. So what I think is happening, I think that there is this amino acid glycine encoded by this codon GGA. I wait a certain amount of time, there is a change. This change is synonymous, it's still glycine. Then I wait again, and then I have a change and this becomes alanine and stays alanine and so forth, right? So I have changes between all the codons randomly, or if I work in the amino acid space, I have 20 states, and this is a Markov process with 20 states and continuous time. So I can fit models into this, into my phylogeny, and estimate what are frequencies of amino acids, stationary frequencies of amino acids, and what is the rate of the process. Another type of information we're trying to use is structural information. And the important observation here, originally published by John Malt, and I regret that we didn't publish it, is that most pathogenic mutations affect stability, not specific functional sites. Which seems surprising, but the idea here is that mutational target for everything which would affect stability is much greater than mutational target for specific catalytic or binding sites. So this seems to be good news. So you can estimate delta delta G, so a change in free energy of folding, and use some biophysical biochemistry methods. However, there are a couple of problems, many problems related to real biology, but the main problem here is that we're very bad at this. So the attempt to estimate delta delta G, to use some methods which would work relatively fast, estimate delta delta G and apply to data, in our hands doesn't work very well. It does work much better than random, but that may not be good enough for your genetic project. So we use a number of heuristic structural parameters that actually help. And we combine all of the sequence parameters and structure parameters, throw this into probabilistic classifier, and generate prediction. So this is our method, Polyphen2. There are a bunch of others that do relatively similar steps. So what is the problem with using comparative genomics and this type of reasoning that somebody variant has to affect structure and also fit the observed pattern of changes during evolution? So the main issue here is this assumption of constant fitness landscape. So what we assume is this. So if you have this allele on this genetic background and it's pathogenic, it's also pathogenic on a different background, and it's pathogenic on a completely different background, right? May not be the case. It may be that this allele is pathogenic here, here, and is not pathogenic here. So on this background is completely neutral. We looked at this. The reason horse is in this picture. About 10 years ago, we had a paper with Alex and Fedor Kondrashevs looking at this issue. And this is structure of human hemoglobin. And you have this at least the database suggests that changing valine in position 20 for glutamic acid is a pathogenic mutation, right? And you look at the human protein, and it sort of makes sense. Glutamic acid wouldn't fit here. Then you check multiple sequence alignment, and horse has glutamic acid in this position. And I'm pretty sure that horse is okay. This horse was fine. And apparently, I believe, runs much faster than I do. You look at the horse protein, and you see that we never tested this experimentally. I'll come to things we did test experimentally, or our collaborators did. There is histidine 69 in this position, which is probably protonated. So this interaction looks stable on the horse background and not stable on the human background. So it is possible that the putative effect of this change depends on the background, on the genetic background of the species. Now we have plenty of vertebrate and plenty of mammalian species, thanks to a lot of colleagues at Broad and other places. And we checked this. We decided to recheck this recently, and this was work by Daniel Jordan in the lab, who is a Harvard Biophysics PhD student. And what we find that even if we're restricted to mammals, you have 5 to 6% of mutations annotated as pathogenic in humans found in this mammalian alignments, right? I don't have time to go through all this debate how many of those are alignment errors, how many of those are database artifacts, and so forth. But at least a certain fraction we were not able to remove all of them. Now, what we think is happening? So what we think is happening is that these variants are pathogenic on the human background and maybe benign on a different genetic background. The problem when we started thinking of that is that these interactions, this can be very complex epistatic interactions. There may be many variants involved in compensating the effect of this pathogenic mutation on a different genetic background. So we decided to look at the models to answer this question, and the first thing we've done, we developed very simple, straightforward models. So say you have variant of interest, and say in the beginning that this variant is completely neutral. The question is, how far shall I go in the phylogeny to find this variance in another species, right? So I go down phylogenetic tree, I go to apes and monkeys and then mammals and vertebrates and so forth. So when for the first time I would encounter the same exact amino acid, right? So statistically, this is a Poisson process. I expect to see exponential distribution, and Daniel looked at the data, and this is actually a pretty nice fit, right? So you go down phylogeny, and this is the distribution of waiting times. Now assume that the mutation is a pathogenic mutation. You do the same plot, it doesn't fit. It's a different plot. Then what may happen? So potentially there are many other changes in the sequence of the same protein or other proteins, and let's call it parameter K, which have to happen out of target of M, and you can fit the model of convolution of these processes, right? And what was interesting is that when we look at the model, the best fit we get is simple gamma function, meaning that there is one change necessary to allow the second change, right? So it's sum of two exponential processes. Now this is model fitting, nobody would believe that, right? But this was enough for us to think, oh, it seems that this modeling predicts that there is one single compensatory event, at least within mammalian or vertebrate tree. So we're not walking too far to find these complex interactions. And at that point, we became brave enough to talk to experimentalists, and very fortunate to start collaboration with Nika Katsanis at Duke. So we look again at the tree, we find these situations where there are potential compensations, there are many, and you can come up with many candidates for this effect of genetic background, right? And I was scared in the beginning because there were many of these variants. Nico said that his lab injects 500 fish a day. I don't know whether this is true, but at least they could pull it off. So this is the idea of the experiment. So this is the type of experiment he does. The phenotype is this angle. This is specifically for one system. They use BBS, Biddle-Bartle syndrome. This is fish phenotype. So you knock out the gene, and this angle becomes much larger. Then you try to rescue this with human mRNA, and there is usually complete or partial rescue, at least in this system. So human mRNA completely displaces and functions instead of fish RNA. Then you try the same with human gene with disease mutation, and if you fail to rescue, this means that the mutation is pathogenic, right? What we suggested of doing is to inject double mutant, and if double mutant rescues, so if, again, knockout produces phenotype, human gene rescues, human mutation doesn't rescue, but double mutant does, this means that this second mutation induces suppression. And the prediction was that the suppression would be complete. So this is how it looks. So you have three classes of phenotypes. Controls are mostly fine. Knockdown mostly are not fine. Rescue with human wild-type gene, it's not fully complete, but it's pretty good. And then you inject double mutants, and they are the same as a single mutant, right? So most of the fish are sick. Then you keep going, and there is one variant which provides complete rescue in the phylogeny, complete suppression, right, in this in vivo system. And one provides partial. Another gene in the same system, again, most double mutants do nothing. One double mutant provides rescue, complete rescue back to the phenotype. Can this work for de novo cases? So this is a patient. She developed a whole bunch of very debilitating conditions. Parents are unaffected, so there were 4 genes were followed in recessive mode and de novo mode. Well, Titin is clearly not functional here. All other genes were tested in the fish, and BTG2 also is a very good biochemical candidate. Microcephaly is reproduced in the fish system. Neuronal expression and pure cell proliferation, cell count. The problem is that this mutation in PGG2 reverses to mammalian ancestral state, right? So it basically is the native amino acid in fish and in many mammals, right? So the variant which is wild type in humans is only found in primates. Again, double mutant experiment, there are 2 sites on the ancestral background which completely suppress the effect of this mutation. So to end up this part of the story, inferring effects of missense variants are very difficult. Methods based on comparative genomics and protein structures have accuracy of about 80%. The major problem what I see currently is dependence of the effect on genetic background, so epistatic interactions, which are not accounted for in any of the evolutionary models we have in hand, and this is the problem. So in remaining, I think 10 minutes or so, I'd like to talk about encoding variants. So I'm back to where I started, right? So I started with the necessity of analyzing variants in this Mendelian world where penetrance is high, functional effect is big, there are experimental systems available, and potentially there are very few rare candidates. And then we discussed this hugely polygenic models for complex traits, and the fact that many of these variants are non-coding and a weak effect. So here the problem is completely different, and for now the problem is even understand what drives this plethora of weak effect non-coding variants. And the idea is, again, importance of regulatory variation. So variation involved in changing gene regulation. There are many annotations available from the ENCODE project, and currently from the Epigenome roadmap projects based on primary cells, and the set of papers I think are scheduled to appear in February. The idea of this project is that you measure epigenomic variables such as chromatin accessibility and chromatin modifications. Of course, these annotations don't tell you specifically about the function. What is the regulatory variant, what gene it regulates, and where, and so forth. But that's good for you, right? Because it's very difficult to imagine annotation which would test binding of every single regulatory factor in the genome with specific functional attributes. So these annotations are broad. They follow these very broad properties that for regulatory variant to function, you need open chromatin, for example, right? Or modified chromatin. So why do we think this is important? Again, there are two reasons. One is purely evolutionary reason, and another one is human genetics reason, so following up genome-wide association studies. In evolutionary reason, there is growing literature trying to fit models of natural selection. So this comes from work by Adam Sipel, looking at within open chromatin information content for positions of in transcription factor binding sites, and looking at correlation of measures of purifying selection. And this, again, this is weak selection. The methods are based on the analysis of human polymorphism and difference between species. On genome-wide association studies, so this is paper from paper by John Stamatianopoulos and Chris Kotsapas here, and I worked on this paper as well. So looking at GWAS hits at different p-value thresholds and looking at the enrichment within this regulatory regions marked by open chromatin. And what's happening here is you see strong enrichment of hits within these regions, but the reason you believe the signal is real is because it's frequently very highly tissue specific or cell type specific, right? So I don't know how many of you are as far from clinical medicine as I am, so I had no idea what QRS duration is, and QRS duration is one of the electrophysiological phenotypes for heart. And when you look at the enrichment, this enrichment is specific to heart tissue and not observed in other tissues. Work by Shomurai Chaudhury's lab, a similar concept. So he takes the peak and shuffles it around and develops a measure based on height of the peak, width of the peak, and the overlap with genetic signal, and runs permutations. So he comes up with statistical method to test for enrichment. And what he sees, again, same story. For LDL cholesterol, he sees enrichment specific to liver. In rheumatoid arthritis, it's T cells. And what I think is most interesting, in diabetes, two tissues light up, adult liver and pancreatic islets. How much of this variation is explained by regulatory function? And I mentioned this highly polygenic model, random effect model, statistical techniques, in this case used by Sasha Gusev and Alkas Price lab, trying to estimate fraction of heritability for 11 traits, how it's partitioned between different functional types, functional categories, right? And what you see here is that some fraction of heritability is due to protein coding regions, and these are all complex traits. Enrichment is the strongest. So if you know that protein coding regions are only 1% of the genome, but they explain 14 times more heritability than their size, right? It's very strong enrichment. However, because they are just 1% of the genome, this amount of heritability is not huge. However, if you look at regions marked by DNA's hypersensitive sites, so regions of open chromatin, where you think regulatory action is happening, the enrichment is 5-fold, but this explains almost 80% of all heritability of complex traits, right? Again, this whole idea of numerous variants of statistically very small effect, changing regulation rather than gene function, seems to get foothold in the psyche of our community, right? So this is what's happening. I think I'm out of time. So for the original part of the talk, this is how great my lab is in general and how we carefully think about the projects. So the original part of the work was mostly work by Daniel Jordan, PhD student in the lab with help of Ivan Adjebe and others, and our collaborators at Duke are Nika Katsanis, Erica Davis, and Stefan Frangakis. Well, thank you very much.